I know.
Yeah.
Okay, sweet, you can help me out later today.
Yeah.
I'll go ahead and just get started. Dano can come in. So today we have Shabani Moeller, and he's a PhD student with the advisor Dr. Elizabeth Van Wormer. Shabani is a fourth-year PhD student here in applied ecology and School of Natural Resources. His research centers on One Health, an interdisciplinary approach connecting human, animal, and ecosystem health. With 20 years of experience as a research laboratory scientist in tropical infectious diseases, he now focuses on ecosystem health, studying the impacts of agrochemicals on human and animal health in Nebraska. So today he will be talking to us about tracking agrochemical contaminants in aquatic and terrestrial environments using tadpoles and songbirds as sentinels. So I'll go ahead and turn it over to you, Shabani.
Yeah, thank you so much. Hello, good afternoon. As you said, my name is Shabani Mola, and I'm a PhD student, and this is my fourth year. You can't believe this was my fourth year, and I was like, oh, the days are going so fast. Yeah. Under supervision of Dr. Elizabeth Van Wormer, and I'm happy to present part of my my results today. Okay, before starting, I would like to provide more background about me. Some people don't know me here. Yeah, back home, I'm from Tanzania, East Africa, the home of the highest peak in Africa, Kilimanjaro. And in Tanzania, I worked as a lab scientist, and I spent part of my life there strengthening the health system. And I was confirming what is circulating that human and animal interface, especially the pathogens. My major contribution in just to strengthen the public health system. And as part of my hobby, I like swimming. I like to listen to soccer news too. And yeah, in Nebraska, I work in the lab with Dr. Van Wormer. And I also worked in some country like in India, in the lab, but I'm happy to be here doing my PhD. I'm also a father, 3Ds, Daniela, Dionys, and David. Daniela is 9, Dionys is 7, and David is 4. I balance study life and parenting. Yeah, so today I'm going to talk about tracking agrochemical contaminants in aquatic and terrestrial ecosystem using tadpoles and songbird sentinel. But let's be on the same ground about some definitions and some word like One Health. So what is One Health? This is a concept that it has been there, a concept that's linked the health of human, animal, and the ecosystem. So according to WHO, OIE, and FAO, they define One Health as an integrated unifying approach that aims to sustainably balance and optimize the health of people, animal, and the ecosystem. So it's actually connecting what is happening human, animal, and in the environment. As you may know, there are diseases that are transferred from animal to human. So if you tackle disease on human alone and leave what is going on in the environment, you won't be solving problems. So this international organization and other people, they decided to structure it. How can we combine this in order to deal with infectious disease of global health issues by linking the health of human, animal, and in the environment too. Yeah, we also know about agrochemicals. We are in Nebraska, corn huskers, where agriculture is the main activity, and so many corns are being produced. And in order to do that, they use agrochemicals in order to increase the yield and to protect the seeds. And there is a chemical called neonicotinoid. Neonicotinoid, it's a nicotine related, but it is a synthetic product that is used in order to protect crops. they use it in a variety of ways, that, in soil, they coat it with grain, sometimes they inject in trees in order to protect against bad insect. And it's widely used. Actually, it is good because it helps protect plants, crops, and we see the benefit of it in Nebraska and other part of the world different from Nebraska. However, these chemicals, they have some effect. They have some effect, especially to non-targeted organisms, including insects. They are toxic because they kill insect by acting on the nervous system of the insect. So they paralyze the insect, and as the result, the insect die. In EU, in Europe, these chemicals are not being used, or it is used by limited, but with some limitation. But in the US, we still use it, and there's some study going on how to ban it, but it is still there. So, yeah, in 2021 here in Nebraska, there was a company called AltEn. This company operated in Mead, Nebraska, which is located in northern part of Lincoln. They used to produce ethanol out of the waste of seed corns treated with agrochemical, including neonicotinoid. It was an innovation, and everyone appreciated what they did because it was nice. But they violated some of the rules and principles. As the result, they polluted the environment. They contaminated the environment around the meat, and the Nebraska Department of Environment and Energy, they did the inspection there, and they found that they violated some of the rule and regulation because the NDE, they are responsible for enforcing the Environmental Pollution Act. So as the result, they closed that company and they proceeded with some lawsuit against the company AltEn Nebraska. So this company, they use waste corn seed treated with agrochemicals to produce ethanol. And yeah, the plant was closed in 2021 because of high contaminants, including clothianidin and thiamethoxam, as you can see, but other legacy chemicals were found at huge contaminant concentration. Yeah, other chemicals like fungicide, tebuconazole, they were also detected on the site, which was far beyond the concentration of this chemical were far beyond the EPA allowable concentration in the environment. That's why they closed this company. After closing this company, before closing, there were so many complaints, including this complaint from Mr. Paula Dines. I can just read very quickly the complaint against this company. We would like to file an official complaint about the byproduct being spread in Sounder County. We believe it may contain pesticide residues which may negatively impact the environment and wildlife. How? Description of the problem: our dogs, 3 dogs, were sick last year. It was in this complaint letter was filed in 2019, was sick last year after being witnessed eating the AltEn ethanol byproduct. The complaint letter keep on saying that this year in 2019, 2 of our dogs, they've become sick with the same symptoms last year, coinciding with the transportation of the byproducts past our properties. So the complaint letter says that when did this occur? Each dog was sick at least twice last spring. April 2018, it took us a while to make link between the illness and the byproduct. I can read to the description of the environment from this letter. What does it say? Beyond the illness, our dogs, our neighbor, reported seeing a disoriented raccoon in close proximity to the byproduct, which now being accumulated in large pile within our surrounding field. And the complaint letter ends by saying that I believe that this could have an impact on the soil, beneficial insects, and waterways, in addition to the local wildlife. I like this letter so much. It looked like it was written by someone who is educated, so they know about the effect of this. What attracted my attention about this letter, which is publicly available. You can scan this and get the whole letter online. It's the way they took initiative to test these chemicals, and file a complaint letter with evidence in hand. I like it so much. So, what are we using tadpoles? As you may have known, we all grew up in the village, some, especially those who grew up in the village. We played with tadpoles, in the water. For me, when in my village there back home in Kigoma, when I see tadpole in water, immediately I know that this water is clean, is safe. See? You can't see tadpole in dirty water, especially back home. Why? Because tadpoles are indicator species, They dwell in aquatic environment, in water, and they are directly exposed to waterborne contaminant. The skin is permeable. Some chemicals can just get inside because they have large pore on the skins and they can accumulate some chemicals. But so they are environmental stress, very sensitive to environment stresses. When something happens in the environment, high temperature, low temperature, sometimes they can be affected. And in Nebraska, we have bullfrog tadpole. I haven't seen bullfrog tadpole in my life. I first saw them here in Nebraska. Can you see? This is a picture of the bullfrog tadpole. It's huge. Look at that. Oh my goodness. I used to see small, small bullfrog, but in Nebraska, I saw large tadpoles. They call them bullfrog tadpoles. Scientifically, Lithobates catespianis. That's the scientific name of this tadpole. So these bullfrogs are invasive tadpole. We don't want them. Why? Because they tend to outcompete native tadpole. But they reproduce so quickly. There are so many. But sometimes you cannot find them, because they look at the clean water, the size of the pond, because they are large. So they eat a lot, they need a lot of nutrients in order to grow to that size. And some studies here conducted by Professor at SNR, they found that these bullfrog tadpole, they can be, they have some element of nematode that they can transmit to other tadpoles. So they are not good. We don't need them, but otherwise they need them. Yeah. So they do have some effect of neonicotinoid in tadpoles, documented various species. No, we've noticed that once these tadpoles are exposed to neonicotinoid, they have negative effect, like delayed growth, disorientation. They become vulnerable to their prey. Why? Because they do, and sometimes they die. See, so even movement, they cannot jump because that thing is like nicotinoid. Can you imagine consuming a large amount of nicotine? For human being, you get disoriented. How about this low animal? So that's what it does to this tadpole. And a lot have been documented, and this is a recent publication by Oliveira. Yeah. So songbird, we like songbird, and they are so beneficial for us in Nebraska, where I've seen documentation of how people pay a lot of money in order to watch birds. See, and these birds, they reflect the exposure, on terrestrial, the environment. And the songbirds, specifically red-winged blackbirds, I like them. I didn't talk about them. I learned about them through this project. And you can easily detect the behavior of a red-winged bird by just looking at the male bird. They can take you to the nest. Why? Because they are more aggressive. See? And another behavior of this bird, they nest nearby the water. So it's very easy to track contamination on terrestrial environment, but also aquatic, because usually they prefer drinking water, nesting nearby the wetland. and marshes, ditches. That's where you can get the nest. But also the breeding season, it's in good time, starting from late April, May, June. That's when they breed, which is comfortable in Nebraska. It's not cold during that time. It's a good time to go in the field and collect some data. Yeah. So also there've been a lot documentation linking the neonicotinoid and bird, especially the American Bird Conservancy. They've documented the abundance, the way the number of birds in Northern America has declined. Why? Because of application of neonicotinoid, and there is a link between abundance of insect. If neonic can kill insect, or this red-winged blackbird, they depend on insect. It means in one way or another, they can be affected. And there are proof for that. There are proof for that. And aside from other environmental factors, habitat loss, deforestation, but also agrochemicals have an impact in contributing to the decline of birds, let's say. And ABC, the American Bird Conservancy, has documentation, and these are some of the news concerning neonicotinoid pesticide, the way they threaten the birds in the US or around the globe. Yeah, so my presentation is part of chapter 1 and chapter 3, where my major objective is to assess the spatial temporal distribution of agrochemical contaminants, neonicotinoid and fungicide in wildlife sentinels living in aquatic and terrestrial environments near the AltEn facility in Nebraska. So yeah, I'm gonna use a bullfrog tadpole, but also native tadpoles. And in terms of birds, red-winged blackbird and brown-headed cowbird. And the study period from 2021 to 2024. And that's me with one of the staff here is known Ferrari, Dennis Ferrari, collecting data in the ponder field. Yeah, so the study area, as I said, we collected our data in Sounder counties, which is located in northern part of Lancaster County, or northern part of Lincoln, Nebraska, and the yellow triangle represent ponds, and the red dots, those are nests, bird nests. Yeah, so the design of our study, it was a cross-sectional observational field study. We did a structure sampling by looking at the ponds that are directly linked with AltEn and some that are not directly linked. And we use deep and seam net in order to collect the tadpoles. Yeah, what we collected in 2021, we collected 43 tadpoles with sample from 5 ponds, and we analyzed 166 pesticide and the transformed product. So what did we found? We found that the southern part of AltEn, the middle part here, is AltEn. The southern part, we have, these are the ponds, South 1 and South 2, there are 2 ponds. And the northern part, N1, N2, and N3, these are ponds located in northern part of Lincoln, of AltEn. And what did we found? The southern part, S2, which is directly linked to AltEn, was highly contaminated by neonicotinoid. It means tadpoles collected from South to S2 had higher level of contamination, including clothianidin, thiamethoxam, and other fungicide, including tebuconazole, iconazole, and we screened 166 agrochemicals. Only 14 were detected, means present. 2 of them were neonicotinoid or transformed product, 8 fungicide and the transformed product. We also found legacy chemicals, which are DDTs. We all know the DDT, but they've been banned, but we found them. And yeah, I forgot to mention that the data here, they are also available online through our partner USGS. So you can also find them online. In terms of the percentage of detection, 7% of that, 7% were found neonicotinoid positive, and 18.6 had thiamethoxam specifically, and then the second one was clothianidin. And in terms of other chemicals or other legacy chemicals, they were like 65.1%. See, and fungicide, especially tebuconazole, was 30.2%. We plan to extend our result for 2022. We have already collected data and submitted the sample. We have some results, but we are now analyzing and they'll be ready for next semester. Maybe next semester we are going to present them the extension 2022, 2023 up to 2024. Yeah. So yeah, in terms of songbirds, for songbird, it was also a cross-sectional study, observational field study. We sampled marshes, ditches, and roadside with different connection to AltEn. And the way we were doing, we were searching nests one on one. And yeah, so the preliminary analysis data for 2021, which were also analyzed, we also analyzed by Abby and other members. We collected 33 egg samples from AltEn, and we screened for 166 pesticides. These 33 eggs, these are the eggs that were submitted for screening, because we collected more than 120 eggs, but for screening, because you cannot screen everything, and it also it implies you should have, financial, you have to have a huge amount of money in order to screen 166 pesticides for more than 20 eggs. So we selected just representative, like 33 eggs, to screen for the presence of pesticide. And what did we found? Yeah, before talking about the finding, this is the way we were searching, and that one there is an undergrad was helping us during the field work on how to search bird nests in Mead, Nebraska. So we did a survey, and the method that we use, it was documented by Bellet Sky, who documented on how to do sampling in red-winged blackbird. If you see like 4 eggs, you're not supposed to pick all the eggs. You just take one. You see, if you see 2 eggs of red-winged, if you take them all, it means the birds won't nest there. They will fly. They will go to somewhere places. So we follow the guideline on how to do sampling in bird eggs, and yeah, it was great actually. So what did we found? Some of the findings, we sampled cowbird and red-winged blackbird, but the number of red-winged blackbird was higher, 49 red-winged blackbird and 15 cowbird eggs. And in terms of the length, because we were also measuring the length and weight of the egg, and it was the mean egg weight was 3.5 centimeter, and the mean was 2.1. Yeah. So, okay. Next. Better sampling, the results of the. What did we see in the better? This is AltEn in the middle, and the large dot around AltEn, it shows the chemical, the pesticide, especially clothianidin, in bird eggs, which shows that closer to AltEn or around AltEn, birds were highly or heavily contaminated with neonicotinoid. In the northern part also of AltEn, there were wet cake. It was also contaminated by neonicotinoid. And also there were along the side, the road, there was some contamination of neonicotinoid. And this graph here shows how neonicotinoid, starting with thiamethoxam, this is a neonicotinoid. It was the level of concentration. In comparison with clothianidin, the level of concentration of thiamethoxam was high, but the range, the maximum, the range was high in clothianidin compared to thiamethoxam. And this in the middle, it is a byproduct. And atrazine, it's fungicide, it was also present. We also found some transformed products of DDT. Yeah. In terms of detection rate, we found that neonic, especially clothianidin, 30.3% of these of eggs were positive for clothianidin, and 24.2% were positive for clothianidin byproduct, and thiamethoxam were 9.02% positive. So what do this mean? It can be there. Do they have any meaning? According to the benchmark set up by the EPA, you see, this concentration is high compared to the benchmark set for birds. See, it is high, which means it is not allowed. It is high. It has gone beyond the benchmark that is supposed to be like considered as safe in bird. In terms of spatial distribution, we found that this is AltEn in the middle, and the red dot shows the way neonicotinoid, especially thiamethoxam, it is contaminated. It is next to AltEn. Triangle represents ponds. We don't see so many agrochemical in the pond. Why? Because this is the data for birds. And the airplane represent the nest where we collected eggs. You can see in the northern part, we see chemicals. Yellow represent clothianidin, and the maximum was 315.4 nanogram per gram, which was highly contaminated. And around AltEn, you can see all the chemicals detected. You can see the green, which is DDT. You can also see, not far from there, you can also see atrazine, which is not a fungicide, it is herbicide, also found nearby this area. We didn't see fungicide in eggs. We saw so many herbicides, which makes sense because the birds, they nest in the grass, and people apply herbicide in order to kill grass. So we didn't see so many fungicide in bird. So yeah, the differences of chemicals at different sites. By using Kruskal-Wallis, we found that AltEn interest, this is an AltEn, the concentration was very high, and comparing to other chemicals, it was statistically significant. You see, the neonicotine, which is in violet color, the concentration of neonicotine was high compared to other class of chemicals. And we extended the sampling from 2021 up to 2024, and the result is being processed. Okay, so as part of discussion, we use Tadpole and Songbird as sentinel organism in order to detect contamination of agrochemicals near AltEn in the Mead, Nebraska. And what we found was that a high level of nicotinoid and fungicide in Tadpole and Songbird. So this is the first study in the US and globally to found high level of agrochemical contamination, especially neonicotinoid in bullfrog. And also in bird song, I can say that this is the first study to detect high level of agrochemical contaminant. And the contamination is linked to AltEn, Nebraska. You see, they did not only pollute the environment, but the chemical were also available for animals to consume. Yeah, so the future direction, we are expanding our study in order to analyze the data for 2022 up to 2024. And for both, we also plan to include native tadpoles because in 2021, we focus more on bullfrog tadpole. In 2022, we plan to extend species, not only bullfrog and other species too, but also we want to look at to what extent this chemical will persist. Was it only in 2021, 2022, 2023, and 2024? So we are continuing to test these agrochemicals in different years and do other statistical analysis, as you may have done. We have a statistician there. So we do correlation, we have to do some correlation, some inferential statistics in order to determine, okay, these chemicals are there. Do they have some effects in tadpoles? What do they mean? So we will do that study later, but also what is the role of abiotic factors, including temperature, humidity? We plan to do that. So take home message, neonicotinoid linked to AltEn, Nebraska did not only pollute the environment, but it was available, it was bioavailable to organism. As we read from the beginning, dogs, raccoon were affected, but this is at higher level. How about at indicator species? You see, so indicator species were affected before it reaches to the higher level. These small animals, they are already dead. So we also witness maternal transfer, eggs and tadpoles. They got this, or they were exposed to neonicotinoid from their mothers. So it's transferred from the mother to exposure to the to fledgling, exposure to the tadpole. So what do we recommend? We recommend continue by remediation, and we should also do further studies, and also the managers, conservation managers should also think about how to include early indicator species when they are doing the management decision. Yeah, so to wind up my presentation, thank you so much for listening to my presentation. I'm also thankful to my committees, including the chair, Dr. Elizabeth Van Woma, Larkin Farrell, Judy, Michelle Hadley, Mary Drosler, and Dennis Ferrari, and Abby, a student, undergrad student who is now in Netherlands. She also did a study on this neonicotinoid. She helped us collect some data. I'm also thankful for my sponsors. This is the Downstream Water for Food. They're the one who are making me, to live in Nebraska. They provide some funding for me and they support my studies. UNMC, these are our collaborators, and the USGS, they help us to analyze the data, screening and analysis, and University of Nebraska Lincoln, specifically SNR, I'm thankful for them because they are admitted and they're providing mentorship for me. So thank you so much for listening.
We have some time for questions, so anyone?
So, well, it's very interesting. So are you finding the way the birds and the tadpoles are getting contaminated? Is it just like the water has the runoff in it and they're like drinking the water or living in the water for the tadpoles?
Thank you so much for a great question. Based on the way we analyze, we conclude that the tadpole are contaminated through a direct path from AltEn through water. Because in an area where an area that was not straight or connected to AltEn. There was no, the level of contamination was low or it wasn't there. So we conclude that water has played a huge role to, to infect or to contaminate the tadpole. For the songbird, we don't have a direct answer, but we stipulate that insect, because they eat insect. And in 2021, the insects were highly affected. Yeah, so we do believe that it's through food, but also the water, because red-winged blackbird, they nest nearby water, marshes and dishes. So we think both.
Thanks, that was pretty cool. I'm kind of generally curious about some of the other herbicides and fungicides. Those seem to be all above like recommended guidelines anywhere.
Yes.
Was this due to the chemical spill, or is this just, background pollution? And if it's background pollution, like, does it really matter that the alto is polluting the environment when you have other people who are doing this all the time anyway? Like, what's kind of the implication of this individual, episodic, pollution compared to, long-term pollution through, agriculture or something like that?
Thank you so much. That's a great question. There are variety of agrochemical contaminants in that area, and linking with the results of the NDE.
Mhm.
I saw that. I wanted to link. They found not only neonic, they also found DDT, which are legacy chemicals. Yeah. So these chemicals, they may have, this is what they found, and the results are publicly available. So you can see not only neonic were detected at the site, even fungicide and other legacy chemicals were detected there. And these chemicals, frankly speaking, they do interact with other chemicals, and they have some effect on human, they have some effect on, tadpoles. And it will be better in future to study the interactive effect of these chemicals. But the neonic, they're like synthetic, newly used, because the legacy chemicals, it says that they've been banned, but we don't know if other people are using them, but it has been banned. People are not using them, but they persist a long time. So for me, I can say that we are supposed to refrain from using persistent organic compound, or persistent organic pollutant POP. Why? Because they can stay for a long time. DDT have been banned for a long time, but if you do screening, you will still find them there, and they do have some effect. Why? Because in the immunology, once a foreign organism is in the body, it can trigger the multiplication of cells. See? And if it is still detected there, it means it's a problem. You see? How to address it is just through studies, maybe some remediation, how to remove them, but we still find legacy chemicals which can have negative effects on organism too. So yeah, we have to address them collectively.
Like, in terms of litigation or responsibility for negative impacts, can AltEnergy say like, yeah, sure, you can see neonicotinoids, but you see all these other pesticides and herbicides that we didn't put out, so how can you be sure that actually our pollution is causing the damage compared to something someone did, 10 decades ago? Do you have any idea of how kind of that is handled in a legal sense, or any like ways of proving that actually no, it was AltEnergy that is causing the environmental damage? The fact that other pollutants are there doesn't matter.
Yes. First of all, we have to deal with that through continued research, and that's why we have to extend the research not only from 2021, 2022, 2023, and even longer than that. If someone is funding, you can still go back and look at the baseline, because I do believe that in agronomy, people have been doing research on agrochemicals in the Mead, Nebraska. So you can collect the baseline data to start with. In terms of neonicotinoid in the environment, this is the first study to detect high level of neonicotinoid in the environment, because most of the studies, they do field work, where they expose the tadpoles in the lab, they expose them to high concentration, you see, and they observe them die, you see. A study conducted in Nebraska or in the nearby Nebraska, it was done by somebody called Campbell in Iowa. They found low level of neonicotinoid next by Nebraska. So it gives me a baseline that before our study or nearby us, the level of these agrochemicals was low. That's one. To move forward, okay, let's see when the level of these agrochemicals were detected to be high. Then we found that it coincided with AltEn operating in Mead, Nebraska in 2021 or 2020, people started to complain before that. So we can collect data for 2022 and compare. For data of 2022, you will see the persistence of the chemical, the legacy chemical. If you see them in the high amount level, you can start to think that neonate is an effect, because we don't see them, but the legacy chemical are still there, they are still persistent. So this is one way to conclude or to say that there was a contribution from AltEn, persistency of these chemicals. Before, you still see them. After, you still see them. But neonate, they are current one, they were not used there. At a certain point, you can see up and down. We do understand that these chemicals, they can stay for a long time. And there is a bioremediation action taken at the AltEn. So we need to do a causative studies, cause and effects in order to conclude. But the levels can give us some a place to study. That's what I can say. Yeah, but I do understand the interactive effect with other chemicals can be a big problem to conclude that neonicotinoid has caused this. Yeah.
Thank you.
Yeah.
Really good talk. So I guess I'm kind of wondering about like species richness between sites. So I know you had kind of your control northern sites and then the southern sites. Did you see that maybe there was greater species richness in the northern sites, and therefore maybe it's only the resilient species that are surviving the southern site? And then I also have a second part of this question. You have samples from like, what was it, 2021, 2024. Are any of those resampling? Like, did you resample the same site? Okay, and then with that, are you seeing that maybe like you had tadpoles like 2021 and then you're seeing less tadpoles in 2024, or maybe the opposite? So
Okay, thank you so much for I will start with the first one, the species richness. The main target was the red-winged blackbird. And the cowbird, they come in because they call it parasitic bird, you see. Once you find the red-winged blackbird, in that nest, you will also see the cowbird egg inside there. But our target was the red-winged blackbird. So in terms of species, we had so many red-winged blackbird compared to cowbird egg. And I haven't done special distribution, which side had many cowbird egg, which side had red-winged blackbird egg. But what we saw, and all these cowbird egg were in cowbird egg, they were in red-winged blackbird nest. You see, that's the way we found, because they are parasitic bird. You see, they nest in other, or they breed in other bird nests. Yeah. So yeah. For the second question, can you remind me that again? The second question?
yeah, I guess like with the tadpoles, are you
Yes.
seeing like maybe an increase in numbers of tadpoles, like
Yes.
from like the initial year you sampled to after, maybe a reduction of those chemicals, or vice versa, that there's like this bioaccumulation and therefore a reduction in abundance? So.
Thank you so much. We have increased the sample size from 42 to more than 42. And what we see different things that's until the data are available, for public, then I'm allowed to announce the result. But we have seen something, and I think next semester you'll be around. I'll present about what we are seeing for 2022, 2023, and 2024. Yeah,
Okay.
but we increase the sample size for both the birds and
Thank you.
I have one more question.
yeah.
Well, two questions. First, do you know how long AltEn was in production before they shut down? Like how many years?
Yeah, before this, there were a lot going on. This company operated there, I can say, for more than 10 or 15 years. They were changing their activities. According to what is documented in, if you read the website of NDE website, officially they started in 2015. Yeah, in 2015, that's when they started to produce ethanol out of seed corn, waste product of seed corn. But if you read in the website and other people, the way they say, they started way long before. And people have been complaining, but that's yeah. So, but officially they started in 2015.
Okay, yeah, I do remember, I think it was a couple years ago, there was an article in NPR about Mead. Because people were complaining about like the odor and like you could see the runoff and water and there was just like giant green mounds of like neon corn
Yeah.
residue.
Mhm.
Yeah, it was pretty intense.
Yeah, it just caught the attention of the media and researchers. That's how we were called, but
Yeah,
the state to conduct a One Health research to determine the effect of those byproducts. And there are people who are working on human side, and they found that human being people were contaminated with neonicotine in the urine, and not also in urine, in hair, and in water also were also contaminated, and they saw something there, and all the results are also available at the UNMC website. You'll find all the results. They are publicly available.
and then have you read any studies that indicate how long the neonicotinoids stay in the environment afterwards? Like is there a threshold?
That's a good question. There are articles, actually the EPA, they are the one who evaluate and provide license, for this product after they're big producing the industries or for the manufacturer. They can say some of the neonicotinoid, they can stay in the environment for more than 1,000 days. 1,000 days.
1,000 days.
Yes.
That's so
Yeah,
pretty.
like clothianidin, 1,000 days in the environment. But thiamethoxam, they can say like for 2 years, 1 to 3 years in the environment. Yeah, but atrazine, it can stay in the environment for 1 year, 280 days, see, in the environment. But as you said, there are also other stressors there, you see, they combine. And that's why we have also to study the temperature can also play a part. You see, when it's hot, they can also activate these chemicals, but also species, the way they deal in order to see this chemical in the body is different.
Interesting.
Okay, so thank you very much. I think this is the first seminar I've come with regards to your work. My first question has to do with you said something about, you said there was a letter complaining about
Yes.
the neonics in the area, and you said they found out that the dogs, the illness in the dogs were linked to is it AltEn or something like that?
Yes.
So my question is, as a statistician, how did they get the link? And in your conclusion, you said that neonics are linked to that. From my expertise, I know that you have to analyze to see if there is an association in there before you can say there is a link or there is a relationship. But you going through the slides with us, I didn't see any analysis with regards to the link. So how did you come up with that?
All right. Thank you so much for the question.
That's my first question.
The second
My second question is, okay, so these tadpoles, were all of them exposed to the agrochemicals, or some were not exposed to? So if they were all exposed, like, okay, so we have cases and controls. So if they're all exposed, then they're all cases. Was there a situation where you had some of the tadpoles not exposed? What were the conditions for which they were not exposed?
Okay, thank you so much for the good question. Let's start with this letter. This is just a part of the letter, it's not the whole letter. And if you want to read the whole letter, you can scan this letter and it will take you there. First of all, it describes the way there is a direct link to with the AltEn Ethanol. I can read here. Our dogs were sick last year after being witnessed eating the AltEn Ethanol plant byproduct. This is the first direct link. And they went on describing about their neighbor. See, also their neighbors, they witnessed the same, the raccoon eating the same byproduct, being disoriented. This guy, what they did, they collected the byproduct from AltEn, they sent to the lab for analysis, and they got the result. And it was analyzed in Iowa. See, that's the first evidence. So, according us now, there is I can go to the spatial distribution. Let's start with this one. Yeah. This is the south part. The southern part, I can say. The northern part and the southern part. This is AltEn. The center is AltEn. How did we link AltEn with Tadpole? First of all, this southern pond, it is called Kaiser Chief Pond. There is a creek, you see, somewhere here, which is directly linked to AltEn. And I had a slide where showing the watershed, you see, because of the time, I removed that slide. So there is a creek linking AltEn area up to this southern, but this south one, it is not directly linked to the path. So that's why we found huge contamination in southern part. Why? Because it directly linked to the AltEn through the water flow. The second linked. The northern parts, it's very hard for water to go up. You see, there were no neonic in Tadpole collected in the northern part. So, okay, why some of the Tadpole, if all Tadpole are exposed, why some of the Tadpole in southern part were negative and others are positive? Okay, that's when it comes on correlation or looking at size, stages, because tadpoles, they grow from one stage to another. The younger tadpoles, according to correlation analysis, which is not a causative analysis, I did that and I concluded that early stage tadpoles, they were exposed to a high contamination level compared to adult one. And this makes sense even, in the way we live. Babies or under 5 kids, they are more exposed to diseases because of their immunity is very low compared to adults. So that's why you can see some of the differences. Another thing, it's all about seasonality, the effect of temperature. You see, it can affect. That's why some of the tadpole may be negative, but some are exposed to the chemical. Talking about beds, I can go down here. For bed, there is a map. Okay, let's start with this map. Yes, this is AltEn. Why do we link with AltEn? Around AltEn, we see high level of neonicotinoid compared to the northern part and some of the southern part. And these are bed are migratory. They can move from one place to another. And the difference between this one and Tadpole is that the birds, they eat insects and they can just travel. And this is a wet cake. This wet cake, it came from AltEn because they were using the leftover of the byproduct as soil conditioning to fertilize the soil and distributing around AltEn. That's why we can see some neonicotinoid close to the wet cake. It is another proof that AltEn or agrochemical linked to AltEn were positive. All this area, in this area, 94% is it's a cropland. We collected eggs to different area, but we couldn't see neonicotinoid to other area, but except AltEn or area close to AltEn. So it gave us the impression that AltEn has a huge link to the bioavailability of this chemical to animals. Yeah, I did some correlation and found there is the correlation, which is not causative, you see, between presence of these chemicals and distance to AltEn. Yeah.
We have 2 minutes left, if anyone has one last question.
Yeah, so maybe we have some question online.
I did look at there, I don't think.
Oh, okay. Yeah, so if anyone has a question, advice, please feel free to reach out so that we can improve. And yeah, we can improve on my studies, chapter 1 and 3.
Awesome. Thank you so much, Shabani. That was great. Thank you all for coming. We'll be back next week. Thanks everyone on Zoom.